Hello everyone. So with the new things emerging and the field of genetics, the treatment gets better and better. One of the recent most discovered thing is CRISPR-Cas9 system. Have you ever wondered that if you would have had a stitch, if you would have had a small breakage in a cloth, what would have you done about it? There is a tear in the cloth. The best thing you would have done is just repair it. Well, can you have that beautiful thing in the gene as well? So that means, would you have had a scissor from which you would have cut the DNA, repaired it, and end of the story? Would that not be the end of all mutational disease that we are currently facing? One of the way of doing this thing is a magic wand called as CRISPR-Cas9 system. To introduce this CRISPR-Cas9 system, let me just give you a basic facts about the CRISPR-Cas9 system, because this is the talk of the town. In October 2020, two scientists, Carpenter and Laudana, they received Nobel Prize in the field of chemistry, and I can assure you by the end of this video, you will not only get a good idea of what exactly CRISPR is, but also will appreciate their beautiful work in the field of genetics. Let's start. What exactly is the system called as CRISPR-Cas9? It is called as CRISPR-Cas9 system. Let me first tell you what exactly does it stand for. So CRISPR stands for clustered. Okay, it stands for clustered regularly. Okay, it stands for clustered regularly interspersed or interspaced, actually interspaced short palindromic repeats. Palindromic repeats. This is what exactly the CRISPR stands for. Well, if you talk about the Cas9 now, the Cas9 exactly stands for the plus. I can say Cas9. So Cas9 will be CRISPR-associated genes. So you would say that this is CRISPR, C-R-I-S-P-R, and the Cas9 would be C-A and S. This is the Cas9 system. So first you must know what exactly does it stand for, and now you will understand what is the system based upon. Well, there is a natural defense mechanism of any bacteria from the bacteriophages. So you know a basic thing that suppose this is a bacteria, this is a prokaryotic bacteria, and this prokaryotic bacteria has this prokaryotic gene like this. Okay, it has a prokaryotic gene like this. Now suppose there is an entry of a bacteriophage like this. There's a bacteriophage entry in this manner. Okay, and this bacteriophage, which exactly is a virus, it may send its DNA into the bacteria like this. Well, when this happens, what happens exactly, that this bacteria can get a bit damaged. It can get a bit damaged by this phage DNA. It can get a bit damaged by this phage DNA. So what can this bacteria do? The bacteria has a unique mechanism. What happens, this phage DNA, it may get incorporated into the bacterial DNA for the first time. The next time when the same phage DNA again comes, this bacterial genome recognizes it. So once it has got hit, the second time it recognizes it. And now what happens, this will produce something called as gRNA, called as guide RNA. The guide RNA, exactly what it does, this guide RNA produces two types of sequences. One is the I can say it's a constant sequences. One is a constant sequence, and one of them is a variable sequence. What happens now, this constant sequence, it binds to this constant sequence. Okay, two types of sequence. The constant sequence binds to the Cas9 system, and the variable region, this binds to the target DNA. In this case, the target DNA being the phage DNA. In this case, it is the phage DNA. Well guys, if I tell you exactly what is Cas9, remember the Cas9 is actually a nuclease system. So Cas9 is a nuclease system. It means it can break the phage DNA. It can cause a break in the phage DNA. So if this happens, what happens, this ultimately the gDNA along with the Cas, what happens in the next step, suppose this is the sequence of the This is the double-stranded DNA of the phage. That is this is a target DNA. It's a target DNA. So what happens? There's a big system called as Cas9 acting on this. There's a big Cas9 acting on it. Okay, it's a Cas9 acting on it, and along with this comes our the guide DNA, the guide I can say guide RNA, and that will bind here, and this will cause a double strand break on the target DNA. In this way, the prokaryotic bacteria can prevent itself from getting damaged by the phage DNA. Now, this is what happens naturally. Can we apply this thing to our own genetic use? Let's see if we can. So in this case, this was a Cas9. Okay, it's a Cas9, and this is the gRNA. Okay, it's a gRNA. To make the things more easier to understand, now I will tell you what exactly happens in the scientific way of using this for our own benefit. So think of what happens. Let's now understand. Suppose I'll now tell you the application based understanding, application of CRISPR. What happens, guys? Let's assume in a way that there's a target DNA. Okay, let's assume this is the target DNA. Okay, this is a target DNA. Now there are many complex things that will happen here, so be sure that you are actually watching this video very carefully. Well, now what happens? Suppose this is the target area. Okay, this is the target area. Okay, the target area, if you want to specially target this DNA, you need to put one type of spacer RNA. So what is a spacer RNA? It's also called a CRRNA, and that CRRNA is the sequence complementary to the target sequence. That means here there is a spacer, and this spacer, also called as the CRRNA, it has to be given along with something called as tracer RNA. So this thing is a tracer RNA. Okay, it's something like a loop like this. Something like a loop like this. So if you just, what will happen? These two are actually complementary, so we'll make a complementary strands among themselves. What I'm trying to say is, what is happening? These two are all making they all are complementary, and they're binding to it. Basically, what you're trying to do is here in this case, you're trying to suppose this one. This is the target DNA. It's a target DNA, the DNA which you want to target. Okay, it's your suggestion as to what DNA you want to choose. Okay, now to do, you take a spacer. This is a spacer. Okay, it's a spacer, and the spacer is actually the spacer is also called as the CRRNA. I'll write everything about it. First, you'll understand this. Now, this thing exactly is a tracer. TRACR, tracer RNA. It's a tracer RNA. You might be wondering, what are these? Let's understand this. Actually, what happens? The spacer, which is the CRRNA in this case, the spacer is actually the sequence. It's a sequence. Okay, it's a sequence complementary. It's a sequence complementary to the target DNA. Okay, it's a CRRNA. Okay, now you must understand what is the tracer RNA. Now understand the tracer RNA, TRACR, tracer RNA. This actually is a short form of it's a short form of trans-activating, activating, so A, okay, A and C, and it is the trans-activating CRISPR. This is not C here, sorry, it's a C here. CRISPR RNA, and hence called as tracer, TRACR. So TRAC and the RNA. Okay, now what does this tracer RNA does? Tracer RNA you have to put. Suppose there is a, let's understand like this, that suppose there is a DNA. Okay, this is a target DNA. Now if you want to target this DNA, you have to put a sequence that will specially go and attach here, right? So if you know this sequence, of the DNA, what you can do is you can target this. To target this, you have to put two things: a combination of CRISPR RNA that in this case is the CRRNA, that is also being called as spacer, Along with this, you have to put a tracer RNA. Okay, so these two in combination, you have to put. These two are basically, you have to insert. Okay, you have to insert these two. That means in these two examples, these two are separate entity being put together, and then they will go and bind to the target RNA. If you understand this point, again, let me just explain to you here again. Dekho, this is one sequence and this one is other thing. And these two are being put together in a gene so that these two ultimately join together, and this holds the two things into the spacer area. And the spacer is complementary to the target RNA. But the big question is, how will they identify the target DNA? Usually, they identify this by a PAM sequence. Now, you might be wondering, what is a PAM sequence? The PAM sequence usually is just upstream of the target RNA. Suppose the PAM sequence is somewhere here. This is the PAM sequence. Now, you might be wondering, what is a PAM sequence? A PAM sequence, guys, PAM sequence, it stands for proto spacer. It's a proto spacer adjacent. It's a proto spacer adjacent motif. Okay, and hence referred to as PAM, PAM, proto spacer adjacent motif. Usually the PAM has a NGG region. It has a NGG region. Again, what is NGG? So mostly, especially in a case of, suppose, a Streptococcus pyogenes, that is the bacteria from which you get a Cas, you actually, it is NGG, where, as you understand, G is the guanosine as a base pair, and N can be any nucleotide, doesn't matter. That means just upstream to the target RNA, there is something called as PAM sequence. So this PAM sequence helps the spacer and the tracer RNA to attach to the target DNA. This target DNA in this case can be also called as, protospacer. These terms are a bit important, guys. Okay. Now what happens when everything is happening here? So with the help of this, spacer, tracer. What happens with the help of the PAM? The spacer and the tracer RNA bind to the target DNA, and what is also happening, with the help of everything comes the Cas9 system. So this Cas9 nuclease will bind to the entire thing like this. This whole thing is now bound by something called as Cas9. So this whole thing is now bound by this nucleus called as Cas9. And Cas9 is, as I told you, it's an endonuclease. Therefore, the Cas9 is capable of breaking it from the center. The Cas9 system induces a double-stranded break in the DNA. Okay, so this, what happened? This thing, whole thing, this is the Cas9. Okay, it's a Cas9 system. Cas9, I told you, it's a CRISPR-associated nuclease. So now what happens? The entire DNA can now be broken into this nick. The entire target DNA is broken down. So the sequence, what happens now, that if this was a target sequence, the target sequence, because of the action of CRISPR-Cas9, Cas9 being an endonuclease, will break this DNA like this. Okay, so as you see here, there is a double-stranded break. There is a double-strand DNA break. There's a double-stranded DNA break. There's a double-stranded DNA break. So to achieve this, you have given what all these things? You have first given a spacer, which is the CRRNA. Along with this, you have given a tracer RNA. Sometimes what happens, the tracer RNA and the CRRNA is combined together, and that is called as single guide RNA. That means sometimes you give a combination of synthetic single guide RNA. So single guide RNA is a combination of two things. It's a combination of two things. First of all, it's a CRRNA, which, as you understood, Actually, the crRNA being a sequence that is I'll say complementary to the target RNA, and along with this you give a tracer, TRACR-RNA, tracer RNA. So this two things in combination is being called as a single guide RNA. Single guide RNA. That means what? That means in the previous example, if you look here in the previous example, these two things were separate, right? You see, this was separate and this was separate. Now, if you combine these two things together, then this will be called as a single guide RNA. That means if I make for you a single guide RNA, how will that look like? It will look something like this. Dekho, if this is a sequence, okay, so what will happen? Suppose there is a target RNA is here. So what will happen? You will have to give a single thing. That's why I'm just making a single color. You will give, this is the sequence, and the sequence has now attachment to this tracer RNA. So both of them are combined. Both of them are combined. That means this will be the crRNA and this one would be the tracer RNA. So when these two things are combined, the whole thing in combination is called as SG, single guide RNA. And ultimately, again the same thing happens. A large Cas9 system, it binds to it and induces a double-stranded break in the whole DNA sequence. So now you are able to achieve a double-stranded break into the DNA. What next? So because you are able to achieve that break, the next thing you should do is repair that. The two ways of repair. The next step should be repair. Repair of what? Repair of double-stranded DNA break. Now remember guys, the two ways of achieving such type of repair. A repair can be done by non-homologous end joining, okay, non-homologous end joining. It is called as NHEJ, or sometimes it can also be achieved by, it can also be done by homologous end joining. Okay, it can also be done by homologous end joining. Now, if you're wondering what is the basic difference between these two, so remember one thing, simple thing, that the non-homologous end joining, this is not a very good way of repairing. It's not a very good way of repairing. So what happens in the non-homologous is, that means, because this is a very simple way. So the DNA break here. Okay, so some proteins will attach here, like the KU70 can attach, a DNA PK can attach. Okay, so KU70, or the protein kinase can attach, and ultimately they can bring upon the attachment like this. They can start repairing these two, but the problem is this type of repair often is having lot of errors. They can have a lot of errors. That means even though they are joined, it is quite possible that some of the base pair can have a mutation here. Some of the base pair, it can be an error. There can be error in repair. Well, if there is an error in repair, the entire gene gets inactivated. It gets inactivated. This causes insertional inactivation of the gene. Well, if you are wanting this to happen, you can allow this to repair by the non-homologous end repair. But sometimes you need to do a homologous repair. So exactly what is homologous repair? Let's tell you again in short. Suppose there's a double-stranded break in of the DNA of the chromosome. Remember, any chromosome has a homologous from the other parent. Suppose, let me talk about the chromosome number 11 of the beta gene of sickle cell anemia. The same chromosome will also be given by the other parent. That means, suppose these two are of the father's chromosome, so the other homologous chromosome will give a template for repairing. Okay, so this one is the homologous, it is the homologous DNA. It will give a template for repair. So this will give a template for repair. The entire thing will repair like this. The entire thing will repair for like this, and this is a very good way of repairing, less error, and usually it is error free. The whole thing repairs in a normal way. This allows a better way of repairing. It's a better way of repairing. Okay, it is error free. Error free. Now listen to me carefully again. Let's assume the cut you made was in the beta gene in a case of sickle cell anemia at the 6th position. Now, if you want this gene to repair by the homologous mechanism, you need to know what exactly was wrong. For example, the guanosine was replaced by cytosine in the mutation. So what you need to give? You need to give a plenty of cytosine here from outside, so that you need to again give the guanosine in the repair mechanism, so that when the entire thing is repairing, the guanosine will again be brought and the entire thing is repaired normally with guanosine only. So this will be error free, and not only error free, this can be now, the whole thing can now be a normal gene without mutation. And this gene, as you understand, can function, it can function normally. This whole thing can function normally. So this CRISPR-Cas9 is just like a scissor. You cut that DNA, you give some base pairs, so that the homologous way of repairing starts, and now the disease is cured. Isn't it a beautiful way of repairing the entire DNA? So a few things that you must understand is this way of mechanism, where the CRISPR-Cas9 is a very, very important role to play. You basically insert in a cell a spacer, that is a cRNA, and also you give a tracer RNA. If you combine these two, it is called as single guide RNA. The single guide RNA, or these two in different places, go and will attach to the target DNA. Obviously this target DNA is complementary to the spacer. It will create a double bond with the help of this Cas9, and now this can be repaired by the non-homologous or the homologous end repair joining. Undoubtedly, this will require a lot of efforts, and therefore two scientists received Nobel Prize for this, who were Sir Charpentier and Donda. They were scientists from Germany and US, and they have discovered this CRISPR, which brings into the way of how a gene can be repaired by genetic mechanisms. I am very sure it will go a lot of ahead in research, and we can now actually get the treatment for any genetic disorders. The time is just to prove this. I am very sure you might have understood this very clearly. Do give your valuable feedbacks in the comment section below, and let me know how did you like the video. Thank you, best wishes, and stay blessed.